You guys remember the big four? If not, this is a good time to wake up your brains to think about this stuff. Way back in the fall term, we talked about the big four biomolecules that make up all living things. And we have carbs, carbohydrates. We have fats, fats and lipids. We have proteins. And we covered these ones significantly in the fall term. And then I waited until now to cover the last one, which is nucleic acids. Now, you guys all know today that nucleic acids are the molecule that's involved in heredity, passing on the traits that your parents have to you, so that's the reason why you look and sometimes even act a lot like your parents. But we're going to do a really thorough investigation of this. We're going to look at the nucleic acid at the molecular level, just like we did with the carbos, fats, and proteins. And but before we do that, it's important that you get a sense of where it is that this DNA is located, and also to know that it's not just DNA. Nucleic acids, there are fundamentally two nucleic acids, and they can be subdivided even further, but we do have the deoxyribonucleic acids, or the DNA, and we have the ribonucleic acids, RNA, and you should also, I don't know if we covered this, I can't remember, but they're made up of building blocks, just like all the other things are made up of building blocks. Carbohydrates are made up of simple sugars, fats are made up of fatty acids and glycerols, proteins are made up of amino acids, while nucleic acids are made up of things called nucleotides. And in fact, there is one nucleotide with which you are very familiar, and that is the nucleotide ATP. Now, it's funny because you don't think of ATP as being a molecule of heredity, and that's because it's not. As we all know, ATP is a molecule that's involved in energy. However, it is still a nucleotide. So that means not all nucleic acids, or what they're made up of, are responsible for doing one thing. They're not just there for heredity. Now, the way that the DNA is organized, so I'm going to focus in on the DNA in particular, is organized in you and me is very different from the way it's organized in prokaryotic organisms, or the bacteria. Now, again, just to remind you. Okay, so what I'm drawing right here is a bacterial cell. Now, keep in mind that a bacterial cell is tiny when compared to your typical eukaryotic cell. Like if I just decided to take the eukaryotic cell out of your liver, let's say, this is what your cell would look like, okay? And a prokaryotic cell, a bacterium, would look something like that tiny little blue speck. So there's a huge difference in size, which means that there's not a lot going on, relatively speaking, inside of a prokaryotic cell, bacterial cell, as compared to a eukaryotic cell. You just can't fit that much stuff into there. So let's, oh, where did it go? What the? Oh, there it is. I skipped 2 pages here. So here's my bacterial cell, which is really tiny. And if I were to make the DNA red, all right, so it's a long molecule, and in this case, the DNA is interesting because notice that I'm going to start here on the left, all right, so I'm going to start over in this area right here, and I'm not picking up my stylus as I do this. I'm just making sort of a big tangled mess of a single strand of DNA, but it's not a strand of DNA because, in fact, it loops right back on itself and reconnects. So if I were to pull out this piece of DNA here, this chromosome, out of this bacterial cell, it would actually be a big loop. Okay, so you could pull it all out and there'd be no beginning or end. It's just a single loop. Now that said, in bacterial cells, there are also, I'm just going to make this pink, so we understand it's slightly different, but it's still made of DNA. We are going to have loops of DNA, and these loops of DNA are not part of the bacterial chromosome. These loops of DNA are pieces of DNA that the bacteria has picked up as it cruises around inside of its environment. And those things are called plasmids. Now, it's very important that you understand that plasmid M, okay, those are the little loops of DNA, as opposed to a plastid. Plastids are like chromoplasts, chloroplasts, and leucoplasts, okay, which are organelles. Plasmids are tiny little pieces of DNA. They're much smaller than the plastids. All right, so notice too that in this in this prokaryotic cell, there's no nucleus. Now that's not news to you. This is something we covered way back in the fall term. However, there is something called the nucleoid. Okay, and the nucleoid is simply the region that's taken up by this chromosome. Okay, excludes the plasmids actually. The plasmids aren't terribly important in terms of their location because they can be found anywhere. But that little dotted line area, the central region of the bacterial cell, is the nucleoid. All right, so how does this differ? Well, let's take a look. Again, don't forget that the big red thing there is one of your cells. So even though I'm going to draw it the exact same size right here, know that it's much bigger than the cell that I just drew moments ago. And we all definitely understand that inside of a eukaryotic cell, we have a nucleus. Oops. That is not what I didn't want to write a C there. Where's my erase? Where's my undo? I haven't done this in a long time. Okay, so there's my nucleus. And let's zoom in on this nucleus and take a look and see what's going on. I'm still going to draw my the DNA in red, but if I were to draw this, because I'm not going to have a ton of room here, so I'm just going to go ahead and start drawing little things. Notice here that as I draw this, I'm not drawing it in a continuous loop. These are single strands. Okay? Single strands of DNA. And in the case of a human being, you'd have 46 of these single strands hanging out inside of the nucleus. Now, that doesn't mean that all nucleic acids are found inside the nucleus, but the DNA in particular, the DNA nucleic acid is found inside the nucleus. The other types of DNA can actually be found outside of the nucleus, inside of the nucleus, all over the place. Now, this is a little bit misleading. So what I want us to do is I'm going to have us focus in on one particular one of these fellas right there, okay? And just like we called the single loop of DNA a chromosome in the bacterial cell, we would also call this single strand that we would find inside the nucleus of a eukaryotic cell, we call this a chromosome as well. However, the chromosomal arrangement is very different. So let's go to, fresh new page here. If I were to look at the chromosome, oh shoot, I have to go meet up with Miss Lovejoy in a couple minutes. Hopefully I can finish this up. If I were to draw out the DNA in a chromosome in a human being or any eukaryotic organism, it would sort of start off looking the same way that it did when I was making the chromosome in the bacterial cell. So I'm going to start down here, okay? And this time I'm going to kind of cruise around here, loop all over the place, whee, and then maybe come back on itself a little bit like that, and up and a while, it's all sort of crazy thing. All right, except this time I'm not going to reconnect it. What that means is if I were to stretch out this DNA molecule, it would be linear. It wouldn't be a loop. It would be one long piece. But it is important that you understand that when it's hanging out inside, most of the time, when it's hanging out inside of your cells, it's all put together like this, kind of like a skein of yarn, all tightly wound up. And in fact, there are little proteins that hold this all together. And these little proteins, which I'm scattering in here, they're in a much more, they're not so randomly scattered here. They're going to be in places that are very specific. But each of these proteins here serve a very important function. These are called histones. And these histones essentially keep your DNA from getting so wickedly tangled that it would be non-functional. So while it may look really tangled, in reality the histones keep some significant order so that it's been folded in a very precise way and held together, sort of tacked together with the histones to keep it from unraveling at the wrong time and becoming just a big old hairy tangled mess. So again, this is I'm zooming in on this chromosome. So that's why you can see the individual sort of the strand of DNA. And if I were to back up, when we were to look at this from a distance, it would look like this dude right here. So we would have 46 of those tangled messes of DNA hanging out inside the nucleus of each one of our cells. Okay, there's one last thing that I want to cover with you guys, which is a concept that we will be looking at again and again, and you'll never forget it. If there's one thing that 15 years from now, if I come up to you and say, what do you remember from biology, you will remember this concept of the central dogma of biology. Now, the central dogma, don't forget, are like rules that fundamentally support everything that you talk about. So we have a lot of dogma in religions. Okay, well, the central dogma of biology is the same way. Were it not for this very important concept, everything else would fall apart. Okay, and that's not to say that it's not true. It absolutely is true. It's not built on faith. It's built on an understanding of what's going on. So with the central dogma, I'm going to state it in sort of a strange way. It's sort of in more symbolic measure rather than doing it with words. So DNA to RNA to proteins. Now that's the classic central dogma. And what this is essentially saying to you is that DNA, the information found in DNA, is read by RNA, and that RNA is then used to create the proteins that make up who you are and how you function. Now, I like to take it a step further, and that's why I'm going to add a red arrow here, which is technically not part of the central dogma. This is more like Chang's central dogma, and that is to add here traits and behaviors. Okay, because that's what those proteins are responsible for doing. They're responsible for the expression of the way you look and the way you behave. And by behave, I don't mean like misbehaving, you're a little brat. I mean behaving in the way that you do things. Like you can reach up and turn off a light. You can run away from a lion that's chasing you. You can pull the hair of your little sister when she steals your book or something like that. Okay, those are behaviors, and that's actually a bratty behavior on top of that. But by behavior, it's up to you. It's important for you to understand that I mean an animal behavior, the things that you do rather than the way that you look. But they all make up your characteristics. So the traits and behaviors are your characteristics that are essentially who you are. All right, so that's the concept of the central dogma, and we're going to continuously revisit that concept from this point forward. Bye.